Don't watch the clock. Do what it does. Keep going. Happy morning, dear students. For the past 3 months, we have a different way of learning through online, which is new to you, not only to you, but also to me. Online learning make us to move a step ahead apart from our classroom situation. The success of education is not only on learning, but also evaluating the knowledge gained. So this month you are going to write your monthly test. We have completed 5 chapters in zoology. Keep that aside. In order to score good marks in your test, you are going to revise the portion for your August monthly test. Today we will revise evaluation question and answers. Today itself read all the answers and write the assignment in your assignment notebook. In chapter 1, reproduction in organisms, the evaluation 1 mark answers are there in your book itself. Take your book ready and mark the answers. So in evaluation, the first 1 to 5 questions or 1 marks that's answered is already in your book itself. Let us revise. In which type of parthenogenesis are only males produce? So you know the answer, or henotoky. Second one is the animals giving birth to young ones are called viviparous. The mode of reproduction in bacteria is by conjugation. In which mode of reproduction variations are seen? Variations are seen in sexual reproduction because male and female gametes are involved in sexual reproduction. And fifth question is the assertion and reasoning question. In that, the first one is assertion is in bee society all the members are diploid except drones. The reason is drones are produced by parthenogenesis. Both the assertion and reason are correct, and or is correct explanation for A. And second one is offerings produced by asexual reproduction are genetically identical to the parent. The reason is asexual reproduction involves only mitosis and no meiosis. Yeah, it's also both the assertion and reason are right, and or is correct and explanation for A. And Roman letter third is the assertion. The viviparous animals give better protection to their offsprings. Reason, they lay their eggs in their safe places of the environment. No, it's wrong. So both A and R are wrong or false. Viviparous animals means they give better protection to the offsprings. They lay their eggs. They won't lay the egg. They give birth to the young ones. So the option is D. The sixth question is, name an organism where cell division is itself a mode of reproduction. Name an organism where cell division is itself a mode of reproduction. It takes place only in the unicellular organisms like amoeba and vorticella. Amoeba and vorticella are reproduced by cell division. Seventh one, name the phenomenon where the female gamete directly develops into a new organism with an avian example. So the phenomenon is the parthenogenesis. Because what is parthenogenesis? The development of egg without the act of fertilization, that is called as parthenogenesis. So in some organisms, the female gamete ovum is directly developed into a complete individual by the physical or chemical induction. So this is called parthenogenesis. So they are asking the avian example. Avian means birds. Example for this is turkey, chicken and pigeon. Eighth one, what is parthenogenesis? Give 2 examples from animals. What is our defined parthenogenesis? The process of development of an egg into a complete individual without fertilization. That is called as parthenogenesis. I repeat, the process of development of an egg into a complete individual without fertilization is called as parthenogenesis. Fertilization means the egg do not fuse with the male gamete. Example, annelid and sea urchin eggs. Ninth question, Which type of reproduction is effective? Asexual or sexual? Why? So sexual reproduction is more effective method of reproduction than the asexual method because sexual reproduction contributes to the evolution of species by adding variation in a population. So sexual reproduction is a effective method because sexual reproduction involves the fusion of male and female gamete. The fusion of male and female gamete produces a variation. So variation contributes more to the evolution. Variation is the raw material for evolution. So sexual reproduction is effective. And 10th one, the unicellular organisms which reproduce by binary fission are considered as immortal justified. Immortal means they won't die. So the most of the unicellular organisms reproduced by cell division. The whole parent body act as a reproductive unit and disappear after new daughter cells are formed. A single cell organisms, it divides into two. So the whole parent act as a reproductive unit. The whole parent converted into the daughter cells. Thus, there is no death of the unicellular organisms. Hence, they are considered as immortal. And 11th one, why is the offspring produced by asexual reproduction referred as clone? Why asexual reproduction is referred as clone? Because asexual reproduction involves single parent. So, single individual can able to produce the offerings in asexual reproductions. So, these offerings are genetically and morphologically similar to the parent. So, they are similar to clone in all aspects. Thus, offerings produced by asexual reproduction are called clones. 12th one, why are the offerings of oviparous animal at a greater risk as compared to the offerings of the viviparous animals? Oviparous means egg laying, viviparous means giving birth. So the oviparous animals laid the fertilized egg in the open environment without any protection. So they lay egg somewhere. So the development of zygote takes place outside the body of the mother. So mother only protects the baby or the offerings. So the development of a zygote takes place outside the body of the mother, whereas in viviparous animals, the development of zygote takes place inside the mother. Thus, offerings of oviparous survival rate is very less when compared to the viviparous animals. So if you understand the concept, it will be very easy for you to read. Okay, 13th one, give reasons for the following. Some organisms like honeybees are parthenogenetic animals. Why? So male honeybee, that is drones, is developed from the unfertilized egg. Normally you know that honeybees produces two types of eggs. One is fertilized egg, another one is the unfertilized egg. This fertilized eggs develops into queen bee and the worker bee, whereas the unfertilized eggs are develops into drones. So unfertilized eggs are called as a parthenogenetic animals. So males develop from the unfertilized egg, so they are called parthenogenetic animals. And second one, a male honeybee has 16 chromosomes, whereas its female has 32 chromosomes. Why? Because male honeybee develop from the unfertilized egg, so it has only 16 chromosomes, whereas in female it is fertilized egg 2N, so it has 32 chromosomes. Develops from fertilized egg. And 14th question, differentiate between the following. Number 1, binary fission in amoeba and multiple fission in plasmodium. So binary fission means it can produce only 2 daughter cells. Bi means 2, it can produce only 2 daughter cells, whereas multiple means many, so they can produce many daughter cells. This binary fission occur in irregular shaped organisms like amoeba. So amoeba is irregular shaped organisms, so binary fission occur in irregular shaped organisms, whereas multiple fission occurs in a regular shaped organisms. There is a definite shape for the organism. And nucleus and cytoplasm divides once in binary fission, but in multiple fission the nucleus and cytoplasm divide multiple times, so that only they can produce different copies of daughter cells. Okay, so these are the difference between binary and multiple. Okay, next one is the budding in yeast and budding in hydra. Bud in yeast is unicellular, so yeast is a unicellular. Bud in yeast is unicellular, whereas bud in hydra is multicellular. bud originate from a small protuberance on the parent body in yeast. In yeast, the bud develops from a small protuberance. சின்னதாக அப்படியே ஒரு if you see அந்த சப்பாத்தி களி பார்த்தோம்னா அதுக்கு மேலே எப்படி ஒரு சின்ன ஒன்று பூ மாதிரி இருக்கும், அது போல இருக்கும். So budding in hydra arises due to repeated mitotic division. So in hydra ஒரு குறிப்பிட்ட இடத்துல repeated mitotic division நடக்கும்போது அப்போ அந்த செல்ஸ் தான் evaginate ஆகி பட்டா உருவாகுது. Okay, and the third point is yeast gets its daughter nuclei and it may or may not separate from the parent body. Yeast gets its daughter nuclei and it may or may not separate, but budding in hydra means these buds are multicellular and they can separate from the parent body. Okay, and third one, regeneration. Difference between the regeneration in lizard and planaria. Regeneration means producing lost parts of the body. Regeneration. So the type of regeneration in lizard is epimorphosis. What is epimorphosis? Replacement of lost body parts occur in lizard. Whereas in planaria, the type of regeneration is a morphallaxis. Morphallaxis, where the whole body grows from a small fragment. So this is the difference between regeneration lizard and planarian. In lizard, it is epimorphosis, but in planarian, it is morphallaxis. And the 15th question is, how is juvenile phase different from reproductive phase? In the life cycle of human, there are 3 different phases. One is juvenile phase, reproductive phase, and senescent phase. So what is the difference between juvenile and reproductive? Juvenile phase is a period between birth to just before reproductive phase. At the time of from birth, just before the reproductive phase. For example, if a girl is attaining the reproductive maturity at 12 years. So from birth up to 11 or 12 years, it is the juvenile phase. Whereas reproductive phase is the actual reproductive phase. This juvenile phase is also known as the primary growth phase or vegetative phase, whereas reproductive phase is also known as secondary growth phase or sexual maturity. And last question in this first lesson is, what is the difference between syngamy and fertilization? Syngamy and fertilization both are same. So syngamy and fertilization are synonyms to one another. So it is a process. What is syngamy? Syngamy is a process of fusion of dissimilar gametes, that is the male and the female gamete. So the same is present in fertilization. It is the process of fusion of dissimilar gametes. But in syngamy occurs in invertebrates or lower grade organisms. Invertebrates or lower grade organisms such as porifera, coelenterata, like that. So whereas in fertilization occurs in higher invertebrates. Higher invertebrates means above starfish and then hemichordata, urochordata. So occur in higher invertebrates and all vertebrates. So these are the evaluation questions in the chapter 1. So you please go through all the questions and write it in your assignment note. Next we move on to the second chapter, human reproduction. In second lesson, human reproduction, we will discuss the 1 marks evaluation. First one, the mature sperm are stored in epididymis. Epididymis is a storage part of male reproductive system. So mature sperms are stored in epididymis. Second one, the male sex hormone testosterone is secreted from Leydig cell. Leydig cell secretes the testosterone. Third one, the glandular accessory organs which produces the largest portion of semen is seminal vesicle. From the name itself you can easily understand. Semens are produced from the seminal vesicle. And fourth one, the male homologue of the female clitoris is. Female clitoris is homologous to the male penis. The site of embryo implantation is the where the embryo gets attached. It is get attached in the uterus. Next one, the fetal membrane that forms the basis of the umbilical cord is allantois. So there are different extraembryonic membrane. So the fetal membrane that forms the basis for this umbilical cord is allantois. The most important hormone in initiating and maintaining the lactation after birth is prolactin hormone. Prolactin. Lactin means milk. So the milk secretion after birth is produced by prolactin. Mammalian egg is a alecital egg and non-cladoic. It does not contain yolk and it does not contain shell. So it is alecital and non-cladoic. The process with which the sperms undergoes before penetrating the ovum is capacitation. So the process which the sperms undergoes before penetrating into the ovum is capacitation. So painful menstruation is termed as dysmenorrhea. So it is a disorder of the menstrual cycle. So the milk secreted by the mammary gland soon after the childbirth is called colostrum. Colostrum is the first milk secreted after the childbirth. Colostrum is rich in which type of antibodies? IgA antibodies. It's a very important question. It is asked in the NEET exam also. So the androgen binding protein is produced by which type of cells? Serotoli cells. Which of the following menstrual irregularities is correctly matched? So it's a very important question, frequently asked question in the question papers. Amenorrhea, absence of menstruation, is correctly matched. And 15th one, find the wrongly matched pair. Which one is wrongly matched? Luteal phase. Luteal phase rise the FSH level. FSH means follicle stimulating level of a level in the body of the female. So FSH level. Next one is the accession and recent questions. So in this first one is in human male, testes are the extra abdominal and lie in the scrotal sacs, and scrotum acts as a thermoregulator. So both A and R are true, and R is the correct explanation of A. Ovulation is the release of ovum from the Graafian follicle. Yes, it's true. It's occurred during the follicular phase of the menstrual cycle. No, it's false. You might have read the menstrual cycle. So when ovulation takes place, it takes place at the day of 14 ovulation period, not in the follicular phase. Next one, the head of the sperm consists of acrosome and mitochondria. So head of sperm consists of acrosome and mitochondria. Mitochondria is not present in the head, but it is present in the neck region. So acrosome contains spiral row of mitochondria. No, it's wrong. So both the statements A and R are false. And 19th question, mention the difference between spermiogenesis and spermatogenesis. Spermiogenesis and spermatogenesis. There is a difference between these two. Spermatogenesis means it is the formation of a haploid spermatozoa from the germinal cells. Production of sperms from germinal cells is called spermatogenesis, whereas spermiogenesis means the formation of differentiation of spermatozoa. Here, differentiation of spermatozoa takes place from the spermatid. In spermatogenesis, it involves the conversion of diploid structures into the haploid structures. The diploid structure, 2n structures, are converted into single n structure, whereas spermiogenesis changes the haploid structure into another haploid structure. Haploid is converted into another haploid in spermiogenesis, whereas in spermatogenesis, the diploid is converted into haploid. The next point is the growth and division takes place in spermatogenesis, whereas reconstruction takes place in spermiogenesis. And then no organ is lost in spermatogenesis, whereas Golgi bodies are lost during spermiogenesis. So these are the differences between spermatogenesis and spermiogenesis. There is a slight difference in these two concepts. Next, at what stage of development or the gametes are formed in the newborn male and female? So at what stage of development? So production and maturation of gametes takes place during their maturity stage. That means after puberty. Next one, expand. So it's very important question. Expand. FSH. FSH means follicle stimulating hormone. LH. LH means luteinizing hormone. So you have to read the spellings. And HCG, human chorionic gonadotrophin. HPL, human placental lactogenic. So you have to expand these things. And the next one, how is polyspermy avoided in humans? Very important 2-mark question. Once fertilization has happened, that means after the entry of the male gamete into the egg, the cortical granules from the cytoplasm of the egg form a barrier called the fertilization membrane around the ovum. This prevents the entry of penetration of other sperms. That's polyspermy is prevented. Once the sperm enters into the egg, only one sperm enters into the egg. So once the sperm enters into the egg, the cortical granules of the ovum forms a barrier that is called as fertilization membrane, which prevents the further entry of other sperms. Next one, what is colostrum? Write its significance. Colostrum means a nutrient-rich fluid produced by the human female immediately after giving birth, and it is loaded with immune growth and tissue repair factors. So this colostrum consists of immune factors, growth factors, and tissue repair factors. So what is its significance? It acts as a natural antimicrobial agent to stimulate the infant's immune system. So this colostrum contains IgA antibodies, which produces antimicrobial agent. Next question. Placenta is endocrine tissue. Justify. So during pregnancy, the placenta acts as a temporary endocrine gland and produces different hormones such as human chorionic gonadotrophin, human chorionic somatotrophin, estrogens and progesterones are produced from this placenta. So endocrine tissue means they can produce the hormones. So this placenta produces these 3 hormones, so they act as a temporary endocrine gland. So if you see the structure of next question, draw the structure, draw a labeled sketch of spermatozoan. So spermatozoa, you have to draw the diagram and you have to mark the parts. So the parts are the head, middle piece and the tail. So the head contains acrosome and nucleus, and it has a short neck, and my middle piece is present in the mitochondria, and the tail is useful for movement. So this is the diagram. You have to practice the diagram in your notebooks and get ready for your test. Next one is what is inhibin? What is inhibin? State its function. Inhibin is a hormone which is secreted by Sertoli cells. Sertoli cells are present inside the seminiferous tubule in male reproductive system. So Sertoli cells secretes inhibin. They are elongated and pyramidal, and it provides nourishment. Nourishment means food to the sperms. Inhibin plays a negative feedback control of sperm production. So it stops the production of sperms. Inhibin, from the name itself you can understand inhibit means stop. So it acts as a negative feedback control of the sperm production. Next is mention the importance of testis in human. So what is the importance of position of testis? So testis is present inside the scrotal sac of skin that hangs outside the abdominal cavity. It provides a temperature 2 to 3 degrees lower than the normal internal body temperature. Normal temperature is 37 degrees. So this scrotal sac produces 2 to 3 degrees lower than the normal body temperature to provide viable sperms. Thus, scrotum acts as a thermoregulator for spermatogenesis. Question will be asked like this. Why scrotum acts as a thermoregulator? அப்படியும் கேட்கலாம். இல்லைனா the importance of position of testis in humans கேட்கலாம். The answer is C. Next one, what is the composition of semen? Semen is a milky white fluid which contains sperms and the seminal plasma. The seminal fluid acts as a transport medium. It provides nutrients, contains some chemicals. It also protects and activates the sperms and also helps in the motility. Motility means movement of the sperms. So semen is nothing but a milky white fluid which contains sperm and seminal plasma. Next is what name the hormones produced from the placenta during pregnancy. So what are the hormones produced from placenta? You know that there are different types of hormones. Human chorionic gonadotrophin, human chorionic somatotrophin, estrogens and progesterones are produced. Next is what is gametogenesis. So gametogenesis means production of formation of gametes. Gametogenesis is the process of formation of gametes. So meiosis plays an important role in the process of gametogenesis. Describe the structure and structure of the human ovum and with a neat labeled diagram. So you see this structure of ovum. You have to draw the diagram, and it also helpful for you to draw in your practicals. So practice drawing the diagrams. So the human ovum is non-cladoic, alecithal and microscopic in nature, and the cytoplasm is called as ooplasm. It consists of nucleus. The ovum is surrounded by 3 coverings. So what are the 3 coverings that surrounds the ovum? Zona pellucida, vitelline membrane and corona radiata. So between this vitelline membrane and zona pellucida is a narrow pre-vitelline space. So you have to write this and you have to draw the diagram. And next one, give the schematic representation of spermatogenesis and oogenesis. So they are asking only the schematic representation. So you can write this alone. Okay, spermatogenesis and oogenesis. So spermatogonia, how it is formed. From the spermatogonia, how the sperms are formed. From the oogonia, how the ovum is formed. So this is enough. So practice drawing this diagram also. And you have to write or draw all these in your assignment notebook. Next is explain the various stages of a menstrual cycle. Explain the various stages of a menstrual cycle. It's a very important 5-mark question. So menstrual cycle means it occurs in 28 days in the human reproductive life, and there are different phases: menstrual phase, follicular phase, ovulatory phase and luteal phase. So menstrual phase means from day 1 to 5. Follicular phase means from day 6 to 13, and 14th day is the ovulatory phase, and 15 to next 28 is the luteal phase or the secretory phase. In that you have to write what is corpus albicans. What is corpus albicans? In the absence of fertilization, this corpus luteum degenerates completely and leaves a scar tissue called corpus albicans. No need to write it polycystic ovary syndrome. So you write up to this corpus albicans. So if possible, you can draw the diagram also. Okay. Next question is what is the role of oxytocin and relaxin in pasteurization and lactation? Explain the role of oxytocin and relaxin in pasteurization and lactation. So what is the role of oxytocin? Oxytocin means oxytocin causes let down reflex. So it helps in the contraction of the uterus. So rapid childbirth. So the actual ejection of milk from the alveoli of the mammary gland, it also helps and stimulates the recently emptied uterus to contract, helping it to return into the normal size. And relaxin helps in childbirth by relaxing the pelvic joints and by dilation of the cervix with continued powerful contraction. And list out the various menstrual disorders. So you have to write all the menstrual disorders starting from amenorrhea, starting from amenorrhea to oligomenorrhea. So you have to write only the key points. So refer the notes I already sent to you. And next one is identify the given image and label the parts A, B, C, D. So this is the structure of ovum. We already know that. And next is the following is the illustration of the sequence of ovarian events takes place in the human female. So in this diagram, they have given the sequence of events takes place in the human female. So they are asking some questions based on this diagram. So identify the figure that illustrates the ovulation. So this represents the ovulative stage of oogenesis, that is ovulation. Second one, name the ovarian hormone and the pituitary hormone that have caused the above mentioned events. The ovarian hormone is the progesterone and the pituitary hormone is the FSH and LH, that is follicle stimulating hormone and the luteinizing hormone. Next one, explain the changes occur in the uterus simultaneously in anticipation. So what are the changes that takes place in the uterus? The endometrium, that means the inner lining of the uterus get thickened and the blood supply to the endometrium increases. The endometrium is the inner thick layer of the uterus and it gets thickened and the blood supply of this endometrium increases, and it is also essential for implantation of the fertilized ovum and other events of pregnancy. Last question is the differences between C and H. C and H means in this diagram. So C, what is C? C is the secondary follicle and H is the corpus luteum. So write the difference between secondary follicle and the corpus luteum. They are asking indirectly. So C is the secondary follicle, H is the corpus luteum. Secondary follicle is surrounded by layers of granulosa cells, whereas corpus luteum is these granulosa cells are absent. In secondary follicles, there are presence of thecal layers, but these thecal layers are absent in corpus luteum. We are going to revise the third chapter, reproductive health. Important book inside question and answers. First, we start with RCH, reproductive health care. There are various tasks carried out by this RCH. The important tasks are creating awareness and providing medical assistance to build a healthy society, introducing the sex education in schools to provide information about adolescents and adolescent-related changes, educating the couples and those in the marriageable age groups about the available birth control methods and family planning norms, creating awareness about care for pregnant women and the importance of breastfeeding, and to identify new methods or to improve upon the existing methods of birth control. What is amniocentesis? Amniocentesis is a prenatal technique used to detect any chromosomal abnormalities in the fetus, and it is being often misused to determine the sex of the fetus. Once the sex of the fetus is known, there may be a chance of female feticide. Hence, this amniocentesis is banned. What is birth control method? The voluntary use of contraceptive procedures to prevent fertilization or prevent implantation of fertilized egg into the uterus is termed as birth control. There are two types of contraceptive methods. They are temporary and permanent birth control. And we will see the contraceptive methods in detail. Natural birth control method is used to prevent meeting of sperms with ovum. There are several types of natural method. The first one is periodic absistence or a rhythm method. So in the method la pathona ovulation 14th day la nadakum menstrual cycle la. So ovum can survive for about 2 days. Ovum vandu 2 days uyiroda irukum and the sperms remain alive for about 72 hours in the female reproductive tract. So coitus should be avoided during this time. So in the time la intercourse avoid panna. So that is called as periodic absistence. So ovulation time la intercourse irukakudadu. Next one, continuous absistence. So it is the simplest method, rombo reliable method to avoid pregnancy, not to have intercourse for a definite period of time. ஒரு 2 வருஷம் 3 வருஷத்துக்கு குழந்தை வேண்டாம் அப்படின்னு சொல்லி தள்ளி போடுறது. So next is the coitus interruptus. So it is the oldest family planning method in which the male partner withdraws his penis before ejaculation, therefore preventing the deposition of the sperms into the female reproductive tract. So it's the coitus interruptus. Next one is the lactational amenorrhea. So menstruation cycle வந்து reassume as early as 6 to 8 weeks from after paturation. குழந்தை பிறந்த ஒரு 6 மாசம் வரைக்கும் periods வராது. So however the reappearance of this normal ovarian cycle may be delayed about 6 months during the breastfeeding. So this delay of ovarian cycle is called as lactational amenorrhea. So இது ஒரு natural method, but it is unreliable form of birth control. So suckling by the baby during the breastfeeding stimulates the pituitary gland to increase the prolactin hormone. So prolactin hormone வந்து increase ஆகிட்டே இருக்கும் in order to increase the milk production. So this high prolactin concentration in the mother's blood prevents the menstrual cycle by suppressing the release of the gonadotropin releasing hormone from the hypothalamus and gonadotropin secretion from the pituitary. So அப்போ milk secretion இருக்கிறதுனால the gonadotropin releasing hormoneன்றது suppress ஆகிடும். Next is the barrier method. So barrier method அப்படின்னா it prevents a barrier from meeting the ovum and the sperm. So இதில் different types of barrier இருக்குது. Chemical barrier, mechanical barrier, hormonal barrier. So chemical barrier means foaming tablets, the melting suppositories, jellies, creams, இதெல்லாம் chemical agents யூஸ் பண்றாங்க to inactivate the sperms in the vagina. And then the mechanical barrier methods la பார்த்தோம்னா condoms யூஸ் பண்றாங்க. Condoms is used by the male, whereas the female uses the diaphragm and the cervical caps. So this condoms என்ன பண்ணும் அப்படின்னு பார்த்தோம்னா it prevents the entry of sperm into the female reproductive tract. So this condom should be discarded after a single use. Condom வந்து safe, it is a safeguard for the user from the AIDS and also from STD, sexually transmitted diseaseல இருந்து AIDSல இருந்தும் protect பண்ணும். Safe sex contactக்கு condoms plays a very important role. This condom is made up of polyurethane, latex and lambskin. Then cervical caps, diaphragm அது பார்க்கலாம். So female reproductive tract cover பண்ணிக்குவாங்க before the coitus in order to prevent the sperm entering into the uterus. So this is the mechanical barrier. Chemical barrier na chemicals agents use பண்ணோம். Mechanical barrier na condoms, diaphragms and cervical caps. Next, hormonal barrier. Hormonal barrier means it prevents the ovaries from releasing the ova. So ovary இருந்து egg release ஆகாமல் தடுக்கும். It thickens the cervical fluid. ஓகேவா. ஸோ ஓரல் கான்ட்ராசெப்டிவ்ஸ் பார்த்தோம்னா நிறைய பில்ஸ், ஸோ மாத்திரைகள் யூஸ் பண்ணுவாங்க. இட் இஸ் யூஸ் டு ப்ரிவெண்ட் த ஓவிலேஷன் பை இன்ஹிபிட்டிங் த செக்ரேஷன் ஆஃப் FSH அண்ட் LH ஹார்மோன். FSH LH ஹார்மோனை ஸ்டாப் பண்ணுவாங்க, ஓவிலேஷன் ஆக விடாமல் தடுக்கிறது. A combined pill is also present. It contains synthetic progesterone and ஆஸ்டிரோஜன். சஹாலி அப்படின்ற ஒரு கான்ட்ராசெப்டிவ் பில் இருக்குது. அது இட் இஸ் ரிசர்ச் இஸ் டன் இன் லக்னோ. India contains a non-steroidal preparations called centrochromen. Next one is the IUDs, intrauterine devices. So the intrauterine devices are inserted by the medical experts in the uterus through the vagina. So in the devices, there are many devices available: copper releasing IUDs, hormone releasing IUD, non-medicated IUDs, etc. First we see copper releasing IUDs. Copper releasing IUD அப்படின்னு பார்க்கும்போது டிஃபர் ஃப்ரம் த ஈச் அதர் பை த அமௌண்ட் ஆஃப் காப்பர். காப்பரை பொறுத்து அதனுடைய இது வேலை செய்யும். ஸோ காப்பர் ஐயூடிஸில் பார்த்தா நிறைய எக்ஸாம்பிள்: CUT 380A, நோவாடி CU7, CUT 380AG, multilode 375, etc. It releases the free copper and copper salts into the uterus. காப்பரை ரிலீஸ் பண்ணும் and suppresses the sperm motility. ஸ்பெர்ம் மூவ் ஆகிறத தடுக்கும். So they can remain in the uterus for even 5 to 10 years. So our next one is the hormone releasing IUD. Hormone releasing IUD are often called as intrauterine systems. So அதில் வந்து progesterone and LNG20. So they increases the viscosity of the cervical mucus, thereby prevents the sperm entering into the cervix. And then non-medicated IUDs. These are the plastics or stainless steel நால செய்யப்பட்டிருக்கும். Lips loop is a double shaped plastic device which is used as a non-medicated IUDs. So question will be asked in this IUDs. IUDலேருந்து கண்டிப்பாக கேள்வி கேட்பாங்க. 1 marks will also be asked. Chooseல கேட்பாங்க. Dash is a copper releasing IUD. Example உங்களுக்கு மாற்றி கொடுத்துருப்பாங்க. So you have to write the answers. And last one is the permanent birth control. Permanent birth control அப்படின்னா இதுக்கு மேலே குழந்தைய வேண்டாம் அப்படின்னு சொல்லிட்டு ப்ரிவெண்ட் பண்ணுறது. ஸோ அதில் வந்து சர்ஜிக்கல் ஸ்டெரிலைசேஷன் மெத்தட் இருக்குது. There are 2 types of method. One is tubicotomy, another one is vasectomy. ஸோ டியூபிகோட்டோமியில் பார்த்தோன்னா ஃபெலோப்பியன் டியூபை கட் பண்ணுவாங்க. ஃபெலோப்பியன் டியூப்னு சொன்னாலே அது ஃபீமேல் தான். ஸோ அப்போ டியூபிகோட்டோமி இஸ் ஸ்டெரிலைசேஷன் மெத்தட் இன் ஃபீமேல். இன் திஸ் ப்ரொசீஜர் a small portion of the fallopian tubes are cut and tied up by the small incision in the abdomen through the vagina. அடுத்தது வெசெக்டோமி அப்படின்னும் போது இட்ஸ் மேல் ஸ்டெரிலைசேஷன் மெத்தட். இன் திஸ் ப்ரொசீஜர் வாஸ்ட் டிஃப்ரென்ஸை கட் பண்ணி டைட் பண்ணுவாங்க அண்ட் ஸ்மால் இன்செஷன் ஆன் த ஸ்கார்டம் டு ப்ரிவெண்ட் த என்ட்ரி ஆஃப் ஸ்பெர்ம் இன் டு த யூரித்ரா. So vasectomy prevents the sperm from heading off to the penis as the discharge has no sperms in it. What is STD? Sexually transmitted diseases. It is also called venereal diseases or reproductive tract infection, or called as sexually transmitted infection, STI. Normally, this STI are transmitted from one person to other person during intimate sexual contact with an infected partner. Infections like HIV, hepatitis B are transmitted sexually as well as by sharing of infusion needles, that is the Veflon, we can call it as Veflon, surgical instruments, etc. with infected people. Blood transfusion or from infected mother to baby. People in the age of 15 to 24 are prone to this infection. There are different types of STI: bacterial, viral, protozoan, and the fungal STI. Let's see one by one. its causative agent and the symptoms. First is the bacterial STI. Gonorrhea is caused by Neisseria gonorrhoeae, which affects the urethra, rectum and throat, and in females the cervix also get affected. Pain and pus discharge in the genital tract and burning sensation during urination occurs. Second is the syphilis. It is also a bacterial STI which is caused by Treponema pallidum. The symptoms takes place in 3 stages. In primary stage, formation of painless ulcers found on the external genitalia. In secondary stage, skin lesions, rashes, swollen joints and fever and hair loss may occur. And in tertiary stage, loss of movement, mental disorder, visual impairment, heart problems, etc. takes place. Next one is the chlamydiasis. It is caused by Chlamydia trichomatis. This trachoma affects the cells of the columnar epithelium in the urinogenital tract, respiratory tract and conjunctiva that is present in the eye. Next one is the lymphogranuloma veruneum. It is caused by Chlamydia trachomatis. It affects the cutaneous, that means the skin or mucosal genital damage, urethritis and endocervictis inside the cervix. Locally harmful ulcerations and genital elephantiasis are the symptoms of this lymphogranuloma veruneum. Next, we can see some of the viral STIs. First is the genital herpes caused by herpes simplex virus, which causes pain during urination, bleeding between periods, swelling in the groin nodes. Next, second one is the genital warts, which is caused by human papilloma virus. It forms tumors on the external genitalia, cervix and perianal regions. Hepatitis B is another viral STI which is caused by hepatitis B virus, which causes fatigue, jaundice, fever, rash and stomach pain. Liver cirrhosis and liver failure occur in the latter stage. Next one is the AIDS, which is caused by HIV, human immunodeficiency virus. The symptoms of AIDS are enlarged lymph nodes, prolonged fever, prolonged diarrhea, weight reduction and night sweating. Next we see the fungal STI, that is candidiasis, which is caused by Candida albicans. It attacks the mouth, throat, intestinal tract and vagina. Vaginal itching, abnormal vaginal discharge and pain during urination. This fungal STI mainly occurs in female. Next one is the protozoan STI, that is trichomoniasis, which is caused by Trichomonas vaginalis. The symptoms are vaginitis, greenish yellow vaginal discharge, itching and burning sensation, urethritis, epididymitis and prostatitis. It may also occur both in male and female. So next we move on to cervical cancer. Cervical cancer is caused by a sexually transmitted virus called human papilloma virus. This human papilloma virus may cause abnormal growth of cervical cells. The most common symptoms of cervical cancer are pelvic pain, increased vaginal discharge, and abnormal vaginal bleeding. The risk factors of cervical cancer include having multiple sexual partners and prolonged use of contraceptive pills. How you can diagnose the cervical cancer? We can diagnose the cervical cancer by pap smear combined with an HPV test. X-ray, CT scan, MRI and PET scan may also be used to determine the stages of cancer. The treatment of cervical cancer include radiation therapy, surgery and chemotherapy. Cervical cancer can be prevented with vaccination. Primary prevention begins with HPV vaccination of girls aged 9 to 13 years before they become sexually active. Healthy diet, avoid using tobacco, preventing early marriages, practicing monogamy, and regular exercise minimize the risk of cervical cancer. We will discuss about infertility and art, that is assisted reproductive technology. First we see what is infertility. Infertility means inability to conceive or inability to produce children even after unprotected sexual behavior is called infertility. That is the inability of man to produce sufficient numbers or quality of sperms to impregnate a woman, or inability of women to become pregnant or maintain a pregnancy. So this infertility may occur both in male and female. So what are the main causes of this infertility? The main causes are tumors formed in the pituitary gland or in the reproductive organs, malformation of cervix or fallopian tubes, and inadequate nutrition before adulthood. Long-term stress also damages the menstrual cycle, which will affect fertility. Ingestion of toxins, that means heavy metal, cadmium, and heavy use of alcohol, tobacco, and marijuana, that is bodhipurul, injuries to the gonads, and also aging, which is the main cause for infertility. Some of the other causes of infertility in men and women are pelvic inflammatory disease, uterine fibroids, and endometriosis. What is endometriosis? Growth of tissues outside the uterus. Endometrium is the inner layer of the uterus. So a tissue grows outside the uterus, that is called as endometriosis. These are the main common causes of infertility in women. Low body fat in women, that is, this is a psychiatric disorder, eating disorder characterized by fear of gaining weight. Undescended testes and swollen veins in scrotum is the cause for infertility in male. And then tight clothing in men may rise the temperature in the scrotum and affect the sperm production. We all know that scrotum acts as a thermoregulator. You should not wear tight clothes. Men should not wear tight clothes. Underdeveloped ovaries or testes. Female may develop antibodies against her partner's sperm. Male may develop an autoimmune response to their own sperms. These are some of the causes for infertility in men and women. So what is meant by Mayer-Rokitansky syndrome? All women are born with ovaries, isn't it? All women are born with ovaries, but some do not have the functional uterus. Uterus is not functional to bear a baby. This condition is called as Mayer-Rokitansky syndrome. Next, we move on to assisted reproductive technology, ART. ART is a collection of procedures which include the handling of gametes or embryos outside the body to achieve a pregnancy. This intrauterine insemination. This procedure is used to treat infertile men with low sperm count. The semen is collected. What is semen? Semen is nothing but group of sperms. So the semen is collected either from the husband or from a healthy donor, and it is introduced into the uterus through the vagina by a catheter. Catheter is nothing but a wire which carries or which transports the sperms into the vagina. After stimulating the ovaries to produce more ova, the sperms swim towards the fallopian tube to fertilize the egg, resulting in normal pregnancy. So this type of art is the intrauterine insemination. Second one is the IVF. It is also called as test tube baby. In this technique, the sperms and eggs are allowed to unite outside the body in a laboratory. So that is called in vitro, outside the body. Once or more fertilized eggs may be transferred into the woman's uterus, where they may implant in the uterine lining and develop. If excess embryos are produced, they can be cryopreserved, that is frozen for future use. IVF was used to treat women with blocked, damaged, or absent fallopian tubes. Today, IVF is used to treat many causes of infertility. There are some basic steps involved in IVF. They are ovarian stimulation, egg retrieval, fertilization, embryo culture, and embryo transfer. How these eggs are retrieved from the ovaries? By giving the hormone injection, that is HCG, human chorionic gonadotropin injection. Egg retrieval is done by minor surgery under general anesthesia using ultrasound guide after 34 to 37 hours of HCG, human chorionic gonadotropin injection. The eggs are then prepared and stripped from the surrounding cells. At the same time, the sperm preparation is done using a special media. After preparing the sperms, the eggs are brought together. 10,000 to 1 lakh motile sperms are needed for each egg for fertilization. So after this fusion of male and female gametes, the zygote is formed. Then the zygote is allowed to divide, that is cleavage is happening to divide to form 8-cell blastomia, and then transferred into the uterus for a successful pregnancy. The transfer of an embryo with more than 8 blastomia stage into the uterus is called embryo transfer technique. Next one is the zygote intrafallopian transfer. As in IVF, the zygote up to 8 blastomia stage is transferred into the fallopian tube by laparoscopy. The zygote continues its natural division and migrates towards the uterus where it gets implanted. Zygote intrafallopian. Inside the fallopian tube, the zygote is inserted. Next one is the intrauterine transfer. Intrauterine. Inside the uterus, the embryos are transferred. Embryo with more than 8 blastomere is inserted into the uterus to complete its further development. In previous cases, in a zygote intrafallopian transfer or IVF, you can able to see that zygote is introduced in the fallopian tube, whereas in this IUT, intrauterine transfer, the embryos are inserted into the uterus directly. Next is gamete intrafallopian transfer, GIFT. Transfer of an ovum collected from the donor into the fallopian tube. In this, the eggs are collected from the ovaries and placed with sperms in one of the fallopian tubes. The eggs are collected and it is placed in the fallopian tube. The zygote travel towards the uterus and get implanted in the inner lining of the uterus. Here, so transferred inside the fallopian tube. That means transfer of ovum collected from the donor into the fallopian tube. Next is intracytoplasmic sperm injection. In this method, only one sperm is injected into the focal point of the egg to fertilize. Only one fertile egg is taken and only one fertile sperm is taken, and this sperm is injected into the particular point of the cytoplasm of the egg. The sperm is carefully injected into the cytoplasm of the egg. Fertilization occurs in 75 to 85% of eggs injected with the sperms. So the zygote is allowed to divide to form an 8-cell blastomere, and then it is transferred to the uterus to develop a protective pregnancy. Next, we will move on to surrogacy. Surrogacy means It is a method of assisted reproduction or agreement whereby a woman agrees to carry a pregnancy for another person, Vadagetai, who will become the newborn child's parent after birth. Though in vitro fertilization, embryos are created in the lab and it is transferred into the surrogate mother's uterus. After childbirth, this surrogate mother, have to hand over the child to the original parents. Next is the male infertility prevention. How will you prevent male infertility? You might have heard this word azoospermia. In previous classes, we discussed about this azoospermia. Azoospermia is defined as absence of spermatozoa in the ejaculated semen. So, if the sperms are not active or present in this semen, so that condition is called as azoospermia. So, this can be treated with the help of microtesticular sperm extraction. Microtesticular sperm extraction. Microsurgical sperm retrieval from the testicles involves a small midline incision in the scrotum. So, scrotum is present outside the abdominal cavity where the testis lies. So, this scrotum is divided, or a small incision is made in the scrotum, and you can able to see one or both testes inside it. So, under the microscope, the seminiferous tubules are dilated. They are produced to small amount of testicular tissues in areas of active sperm production and can be removed and improved for sperm yield compared to traditional biopsy technique. Biopsy is nothing but taking a part of skin from our body and testing it if there is any problem in our skin and all. Likewise, a small part of testis is removed and it can be tested whether they contain sperms or not. So these are some of the different methods of art assisted reproductive technology. So after this after the mother is pregnant, how will you detect the fetal disorders during the early pregnancy? We have to detect these disorders and we have to correct it. And there are some of the methods for detection of the fetal disorders. They are ultrasound scanning. Ultrasound scanning has no risk other than the mild discomfort due to pressure from the transducer that is used on the abdomen of the mother. So radiations cannot be passed into this mother, and it will not affect the child. So ultrasonography is usually performed in the first trimester for dating. For dating means what? When the child will born, or the determination of the number of fetuses, whether it may be single fetus or it may be two babies. Okay, and for assessment of early pregnancies, if there is any problem in early pregnancies. So this can be find out with help of this ultrasound scanning. It's a normal scanning done for the pregnant women in the first trimester. Okay, so we move on to amniocentesis. This also you might have studied in a previous classes. Amniocentesis involves taking a small sample of amniotic fluid that surrounds the fetus to diagnose for chromosomal abnormalities. So this amniocentesis is also used to find out the sex of the fetus. So this is banned, but it can be used for some time to know about the chromosomal abnormalities in the fetus. Amniocentesis is generally performed in a pregnant woman between the 15th to 20th week of pregnancy by inserting a long, thin needle through the abdomen into the amniotic sac to withdraw a small sample of amniotic fluid. The amniotic fluid is withdrawn from the amniotic sac through a small needle that you might have seen in this picture. So the amniotic fluid contains the cells shed from the fetus. So from the cells, you can able to identify the chromosomal abnormalities found in the child. Next is the chorionic villus sampling, CVS. This CVS is a prenatal test that involves taking a sample of placental tissue to test for chromosomal abnormalities. The placental tissues are taken for a test because the placenta connects the mother and the fetus. So this can be taken for test, and we can find out some of the chromosomal abnormalities with the help of this placenta. Next, we move on to the fetoscope. Fetoscope is used to monitor the fetal heart rate and other functions during the late pregnancy and labor. So we can find out the heartbeat of the child with the help of this fetoscope. The average fetal heart rate is between 120 and 160 beats per minute. But for normal man, it is, that 72 beats per minute. But for our fetus, it is 120 to 160 beats per minute. An abnormal fetal heart rate may mean that the fetus is not getting enough oxygen, and it indicates other problems also.